All right. To introduce the concept of virus evolution, which is, I think, the big takeaway for the entire course, I wanted to start with two smaller concepts. One is optimization, which is the process of producing more of something. Yeah, just sort of changing parameters a little bit until the process works better. The second is optimism. Yeah, the hope that things will continue to get better in the future. And we can look at this from two perspectives, the human perspective and the virus perspective, because, yeah, we got to try and understand the world through their eyes, even though they do not have eyes at all. So let's start off with optimization. So optimization, there are a lot of ways you can look at that. And optimization means different things. It's very context dependent. The optimal runner is probably not the optimal swimmer. I mean, if you look at the Olympics, those gold medals are generally held by other people. And neither of those is probably the optimal business person. Neither of those is the optimal scientist, as far as I'm aware, anyway. And so, yeah, there are different things you can be good at. And so for each of these things, each of these measures, there's a scale and there's some kind of gradient. And yeah, some are better, some are not as good. That's okay. Yeah. I think part of going to university is figuring out how to optimize yourself for whatever kind of future you want. So yeah, we're all in this game together, viruses and people too. And the happy little jumping water glass. As you can see, it's happy, half full, and very happy to be half full up there. Okay. One type of optimization is shown over here. And so this is codon optimization, C-O-D-O-N. And so what do we mean by that? Well, as you probably know, there are 64 total codons. That's combinations of 3 nucleotides, which can be read by a ribosome. And one of those tells us to start, 3 of those tell the ribosome to stop translating, and all the rest of them put in 1 amino acid. Now, there are 64 codons, but there are only 20 common amino acids. And so that tells you that there must be some redundancy built in, meaning that for most amino acids, there's more than one way, more than one codon that will actually code for that amino acid. Shown over here is the example for the amino acid leucine, which has a single letter code of L. So if we look in humans at usage of leucine, the statistics are shown right over here. So there are 6 codons, all of which will bind to a special tRNA, and each of those 6 tRNAs will carry a leucine amino acid on the top of it. So any of those tRNAs can code for a leucine in a protein. And if you have a protein that needs leucine, there are 6 possible solutions that will take care of that for you. Now, if you look over here at usage, you see that some of these, like CUG, are used very often, nearly half the time, 43% of the time. When a protein from a human needs a leucine in it, it's going to use CUG as the codon to get that. If you look over here, things like UUA could totally make a leucine appear in a protein, but this is one of the least used codons. Isn't that interesting? And you can see part of the reason why over here. So each of these corresponds to a particular tRNA. In many cases, we will have multiple copies of the tRNA gene. And so having multiple copies means that we'll make more of these particular tRNAs that bind to CUU or CUA, something like that. But also, there are some of these that are that have better promoters and are expressed to a higher level, or are going to be more likely to be have an amino acid attached to them. Amino acylated is our fancy word. And so those are just going to be more abundant. There's going to be more of those particular tRNAs. And so if you look at tRNA abundance, it's over here. So again, nearly half of the tRNA you would find in a standard human cell is going to be for CUG. So that actually is a good match for the nearly half of the codons which require it. Yeah, the human being is more or less optimized. Look though at CUA. This one is very rare. Less than 5% of the total tRNAs that can encode a lysine are going to be against the CUA codon. Yeah. So, stuff like this is interesting. So the question comes up, well, are viruses optimized? And the answer is that, yeah, viruses are usually fairly well optimized, or at least they're trying to get there for whatever situation they're currently in. And if a virus is not optimized, give it time and it will become optimized, because viruses evolve and the more optimal versions will have a selective advantage, and so they can grow more, produce more offspring, and will eventually wipe out all of the less optimized versions of the virus. Now, here's the problem. So to non-virologists, they generally think, okay, the virus will optimize itself, and they imagine the ways that they think that the virus will need to optimize itself. Like, obviously, the virus will want to keep its host alive, things like that. But of course, if a virus comes in and spreads to a new host, long before it actually kills that host, if the disease takes a month, but spread happens in, a couple of minutes, and then you spread it again within a couple of hours, then there's really no selective pressure that would be trying to keep that host alive, because it wouldn't really matter to virus fitness whether or not the host lives or dies, because once a host has had the virus, it's going to develop an immune response to that virus. And so generally, the virus will not be able to infect that host again, at least not for a while. And so, frankly, any host that a virus leaves is kind of dead to that virus. Yeah, in a metaphorical sense. And so it doesn't really matter if they're also dead or alive in the literal sense as far as virus fitness goes. But that's another story for a little later in this lecture segment. So, some people in the lab of Eckhard Wimmer, who's a noted polio biologist, decided to test this out. They said, well, what is a virus? Is a virus codon optimized for its host, because it's host ribosomes and a host cell cytoplasm that's going to supply the tRNAs and all the translational machinery to replicate the virus, or at least produce its proteins? Or is the virus not optimized? Is it more optimized? Does it use more abundant codons more frequently? Does it use less abundant codons less frequently? Great question. So, if we look at human genes in general, we find that human genes are actually somewhat optimized. So on this particular scale, this is optimization. So positive numbers mean that a gene is using preferentially tRNAs that are going to be more abundant. More abundant tRNAs should result in faster translation, and faster translation should potentially result in more translation. If you imagine that an RNA has a certain amount of time docked up to the ribosome, and it can get copied however many times it gets copied in that time, using the faster, more abundant tRNAs should make the entire process more efficient, and maybe you get 2 copies out instead of 1. Yeah, something like that. So looking at human genes in general, they're about 10% more optimized than we would expect by random chance. So they tend to use those high-frequency codons, and they tend to avoid the low-frequency codons. If you look at poliovirus, it's pretty near dead center. So 0 is neither optimized nor de-optimized. I think it's about 2 or 3% de-optimized, but no big deal. Certainly within the range of a lot of human genes. And so the question they asked was, what happens if we optimize poliovirus, and what happens if we de-optimize poliovirus? Now, these are smart poliobiologists, so they knew that there's a cis-acting replication element that's present in the virus. And so in the case of polio, it's in the 2C gene right here. And so they knew that there are probably other elements like this in there. And so what they decided was they're not going to mess with the P2 or P3 region at all. They are only going to mess with the P1 region, which is up here. And they're not going to mess with the non-coding region because if you mess up the sequence of the IRES only exists at the level of RNA. It doesn't encode a protein. And so if you're just changing the RNA, chances are it's not going to make that shape anymore, and you're not going to work like an IRES anymore. Yeah. So they selected the structural region to change, VP 1 through 4, right there. So let's have a look. So here's our structural region, our P1, 2, and 3. So they tried optimizing all 3 genes. They tried de-optimizing all 3 genes. They tried de-optimizing single genes and pairs of genes. And so, yeah, the question is, what do you think happened and why? Yeah. And it kind of goes back to the question of, was the virus optimized or not? Because the virus, as you can see, is not codon optimized, but viruses do undergo more natural selection more quickly than pretty much anything else. And so you'd expect it to be optimized for producing another infectious poliovirus virion under the conditions in which it normally has to produce new poliovirus virions. Yeah. And if that sounds like almost a non-statement, it's intentional. This is what virus fitness means. It's whatever leads to more of that particular type of virus over time under whatever conditions that virus normally faces for growth. And that's the thing that's really difficult to model in the laboratory, because there's no virus that spreads quite the way that they spread in the laboratory, because we put the virus onto the cells in the laboratory and we close it off, and we usually don't let any other viruses in there. And it's all very carefully controlled. It's not a random mixture of cells. There's no genetic difference from person to person in most cases. It's a homogeneous population of cancer cells that the virus is usually infecting in the lab. And so it is very difficult to draw any real meaningful conclusions from that type of lab experiment that will tell you every single time whether or not a strain has greater potential to cause disease. But let's look at this, because it's interesting. So to understand this, we need to understand a plaque assay. Plaque assay starts by making serial dilutions of a solution containing some virus. Yeah. And we don't know how much is there. We're making the dilutions because we can read out the assay when our number is somewhere between 3 and 30 viruses that are infectious in that sample. And we have no idea how many we start with. So we take the original version, because maybe that'll have 3 to 30, or we dilute it, because maybe that's 3 million. So we have to dilute it once, tenfold dilution, so we'll go down to 300,000. Again down to 30,000, again down to 3,000, again down to 300, and again down to 30, and then we'll be able to read it. So we're going to make these dilutions, and we're going to plate all of the dilutions. We're just going to hope that one of these somewhere is going to give us an answer that we can read. And if not, we have to redo the experiment using either more concentrated or less concentrated virus in order to detect it. Plaque assays tell you how many infectious virus particles are present. They do not tell you how many non-infectious virus particles are present. And there can often be a big difference. For most viruses, like the most well-behaved viruses, about 1 out of every 10 virions will actually be capable of starting an infection under standard laboratory conditions. Now, it may be that in an animal, in a human lung or something, it may be that a higher number can start infections, or it may be that a lower number can. It's hard to predict. Yeah, probably impossible to predict, at least accurately at this point. For viruses like SARS-CoV-2, normally the number is somewhere between 300 and 3,000 broken, dead, non-functional viruses for every one that is actually capable of starting an infection. Seems high, but that's a fairly typical number for this type of experiment in cell culture in a laboratory. So, yeah, kind of inefficient. Oh, well. But, of course, the laboratory is not exactly the same as reality. Yeah, it's actually fairly different. So, You got to be mindful of what you're testing and what your assumptions are behind that. Anyway, to do the next step of the plaque assay, you grow a bunch of cells. And so we're going to grow what we call a monolayer, M-O-N-O-L-A-Y-E-R, which means it's one cell thick and it covers the entire surface of the plate. And most cell types are adherent. They're used to sticking to each other and sticking to basal lamina and sticking to whatever, something. And so you can prepare a little culture dish or plate in such a way that the cell will prefer to stick to it. And so you can grow a little monolayer. And then you're going to put on your virus mixtures. And so some of the virus mixtures may produce a lot of infection. Others will produce only a few infections. Between those, we should be able to figure out how much infectious virus there was. So what we're doing is putting these viruses on, and then we're putting on a thing called an overlay. It's going to be made of agarose. This is the, gelatin-like substance that you get from seaweed. It makes nice little candies and things. We put that on there so that the virus, when it comes out of one cell that it infected, it will then only be able to infect cells that are nearby. So first we put on the virus, let it infect, let it sit there for like an hour, and then we'll come in and put the overlay on top so that the virus can't move around and jump from one cell to another cell that's far away. It can only spread to cells that are nearby. And over time, these cells that are nearby will start to get sick and will start to get infected, and then will start to die. And so you'll end up with a little sort of circle of dead cells, which is kind of like a little crater that the virus has left behind, and we call those plaques. And you just count up the number of plaques. If there's 4 little plaques here, that tells you you had 4 infectious virus particles in that particular solution. Yeah. And so on. And you repeat these things many times because there's a certain amount of error involved. All right, so on to the results of this interesting experiment. What did they find? Well, the virus that is optimized, they called poliovirus max. Yeah, PV max. And so poliovirus optimized, or PV max, is this one with the black circles. And the sort of square right here is poliovirus wild type. So that's where they did not either optimize or de-optimize anything. That's our black bar up here. And you can see those two grew exactly the same rate throughout the entire course of infection. So they are essentially absolutely equivalent little viruses. And they grew it once, then they put it onto new cells and grew it again, just to see if these differences replicated themselves. And so you can see basically they're crossing the finish line in the same order. So white square and black circle, which are our maximum and wild type, are always at the top. And at the bottom is always this triangle, which is PV min Z, which is this one. So we took the third region here, and I don't remember if that's VP1, 2, or 3, and de-optimized just that one, and that seems to hit the virus the hardest out of all of those. Okay, so that's the worst, and the others are in their same rank order each time. So they range from very efficient virus growth to much less efficient. What does that do on the plaque assay? Because people have used plaque assays as a measure of potential to cause disease. In a larger plaque is the result of greater virus spread and greater virus killing of cells. So if you have a virus that is more virulent, that's virulent, it should produce a larger plaque. And if you have a virus that is faster at spreading, or better able to spread, you should produce a larger plaque. It should be able to infect more cells in the same amount of time. So these are all plaques that were given the same amount of time to grow. And you can see for the wild type and the optimized, they're actually about the same. The PV max may be slightly larger plaques. Min XY and Min Z, look at those. And they are tiny little plaques, very, very small. So you grow less virus, and if you put the virus in there, like in a little plaque, you can see that it actually grows less and causes less, a smaller circle of destruction during the assay. So yeah, that is definitely less virus growth. What they then looked at was whether or not this could be a good vaccine. And they concluded that probably this could be a very good way of making vaccines. Because, think about it. You have a virus that you've changed so that it is less able to grow. That's attenuation. The attenuated virus produces proteins. Now, ordinarily, with attenuation, you have to put in mutations, which will change the proteins. And the proteins are what your immune system is going to look at in order to try to recognize the virus and, recognize infected cells. If you have a mutated version of the virus, it may have slightly different proteins. And so your immune system will learn to recognize something a little bit different from the real thing. Sometimes that's okay. Sometimes that can be a problem. Yeah, it just all depends which virus we're talking about and which vaccine. But the beauty of these de-optimized viruses is that the de-optimized viruses encode exactly the same protein. So they look the same to the immune system, and yet they don't grow as well. So you can attenuate something without having to actually change the protein sequence of the thing that you're making, which is kind of interesting, I think. Yeah. So yeah, they called this, the phrase was death by 1,000 cuts, or I think attenuation by 1,000 cuts is a little more appropriate. But yeah, there you go. It's kind of interesting. So what this tells me, I think, is that compare poliovirus, which was right about in the middle of the scale, right about at 0, to optimized poliovirus, which is about 20% ahead, so more optimized than most human genes. And what they find is that there is no real advantage. And so I think the reason why we don't see fully optimized poliovirus is that it's not better. It's not better for producing more polioviruses. Or rather, you can look at it as saying that translation at the level of tRNA selection probably wasn't the thing that was holding poliovirus back at this particular moment. It's probably something else. Interaction with other cytoplasmic components, or, interaction with the cell or something like that. Yeah. But yeah, not tRNA selection. However, slowing down the process does seem to have really bad effects for the virus. So these inefficiently translated parts of the protein, the polyprotein, absolutely beat up the virus and cause it to grow much less well than the original. And the neat thing is that you could put this in, and even if a person was infected with real wild-type poliovirus at the same time, it wouldn't be able to recombine in a way that produced a more dangerous virus. Because it's not just 1 or 2 mutations that are weakening this thing. Every single codon is de-optimized in the entire protein. And so really there's no part that would be similar enough that you could actually get recombination. And if you did, you would be recombining in a defective version of the gene. Yeah, so it couldn't be worse than wild-type virus. So yeah, it's a kind of ingenious little solution. All right. So, with that in mind, let's go over here and talk about disease. So, adaptation is the process of a virus growing more copies of itself in a host. And given time, viruses are really good at adapting to grow in a new host. So you'll have a chance event. You'll have, a chicken sneezing on a person working on a farm, something like that. And maybe the person catches the chicken's version of, bird flu virus or something like that. Probably that virus is adapted to grow in a chicken. Now, chickens have a higher body temperature. They have lots of differences in the protein components of their cells. So if a virus is optimized for growth in chickens, there's no way it can be optimized for growth in people. And so the early viruses that make that jump are probably going to be a little weird. They're probably going to be in one of these sort of fitness valleys where they would probably be kind of bad at growing in chickens, but also kind of bad at growing in people, but just good enough at both of these things that they're able to jump from one to the other. So you got to hit this sort of Goldilocks position, and there's no real way of predicting where this is without just seeing what happens in nature, essentially. Yeah. But then, once a virus gets in, if it's able to spread from person to person to person to person to person, the way that SARS-CoV-2 did, then what you will see is that you will get adaptation. And so the virus will start to grow better and better and better and better and better in the host. And you can see this in some of the viruses. The early strains did not produce as much virus inside of a person or inside of cell culture compared to the later viruses. They're actually producing far more virus inside of people. Generally, you can also model that in cell culture. They'll produce more in cell culture. The question, though, is virulence. So virulence, virulence, is the potential to cause disease. And virulence is unpredictable. Yeah, or it's fundamentally unpredictable what will happen to virulence over time. So a few things that we know. If there is more virus present, generally, usually, there will be greater virulence. Those two normally are correlated to each other. But it's not guaranteed. And there are viruses. One of them is Torquetino virus. It grows to very high levels in people and doesn't appear to cause disease. That's kind of a weird exception to the rule, but it exists in this world, and so we have to take account of it. I think there are a lot of people that would like to see the virus adapt to a new host, to maybe grow better in a new host, but somehow adapt to become less virulent in the host. And that could happen, but there's no guarantee that it would happen. And in fact, I think it's not even the most likely thing to happen, for the reason that we talked about earlier. Because there's only selection when there's a selective pressure. So there would have to be some advantage for a virus that grows in a host and doesn't kill the host. Some sort of long-term advantage in the number of new infections that it starts, or the long-term prognosis of this virus growing in this host or some other host. And that's the thing that I don't think there's any real solid evidence that that's the case. So over time, virulence can rise and virulence can fall. Virulence is going to be a product of both the host genetics and the virus genetics. And without a clear picture of both of those, it's very difficult to predict. There's no real test that will 100% of the time tell you how virulent a particular virus is. There's no test you can do in a lab. You have to observe this in reality. And the problem out there in the wider world is that everybody has somewhat different genetics. You never get infection of a homogeneous population. We don't have too many cloned humans walking around. So it's very difficult to actually say whether a particular virus, variant, strain, whatever, is meaningfully different in its disease-causing potential. These are laborious experiments. They take months or years and tons of statistics and usually millions of data points in order to come to some kind of conclusion. And often the conclusion is that we can't really tell the difference. Yeah. So there you go. So I think that leads us here. So optimism versus pessimism. Over time, should we expect things to get better or worse as regards viruses? I think there's a philosophical difference here, and honestly, optimism feels a lot better. But there are cases with viruses where optimism is not a big driving factor in how they grow. And so I don't think it really matters how you feel about things. And evolution is fundamentally unpredictable. You can predict adaptation to some extent, that a virus will adapt to its situation if it is able to. But beyond that, whether it adapts to cause more or less disease is pretty much totally up to chance. Up to chance and a whole bunch of factors that are, at least at present, probably outside our ability to actually track them and understand them. Because, compare you and me or you and anybody else, and there are differences. We grew up in different places. We ate something different for dinner last night. Different age, different genetic makeup, different T cells, different ratio of different types of T cells. And these are all population statistics. It will also depend exactly which cell the virus hits, where it is in the cell cycle, where it is in the body, whether or not there happens to be a nearby immune cell that already knows how to recognize it, that can shut it down. Just a lot of variables that we really don't have a way of tracking or controlling at the moment. So there you go. Optimism versus pessimism. I don't know. I think neither one of them really gets you very far. You pretty much need to study nature, see what it does, and yeah, that's reality. And how you feel about it is nice, but ultimately kind of pointless. Yeah, there you go. So that's fine. I think that's good enough for this particular section. I don't think there's any more slides in this. Yeah, that's the end of this slide set. So there you go. All right. Thank you very much.